Hello, my name is Braedon Melton, and I've been working in Dr. Beth Conway's lab, and have been interested in studying neprilysin expression in breast cancer and the potential mechanism by which neprilysin interacts with the PI3K pathway in breast cancer. Breast cancer is the second most common type of cancer in women, making it a very prominent research topic, and research done by past students in this lab have shown that neprilysin levels are underexpressed in breast cancer cells and may play a role in regulating the PI3K pathway, specifically through interaction with PTEN. This was determined by pathway analysis software showing that PI3 was significantly upregulated in breast cancer cells. I wanted to further investigate if levels of neprilysin in breast cancer cells were correlated with PTEN levels. Neprilysin is a membrane metalloendopeptidase that has been shown in this lab to regulate breast cancer through cleavage of endothelin 1. It has also been demonstrated that overexpression of neprilysin results in lower levels of PTEN in breast cancer cells. PTEN is a phosphatase that acts as the primary inhibitor of the PI3K AKT signaling pathway. And since correlation between neprilysin and PTEN has been hypothesized in the past, I wanted to further this research. To test my hypothesis that neprilysin is a negative regulator of PTEN, I isolated RNA from breast cancer cells and performed qRT-PCR. The graph shown in figure 1A is a logarithmic graph depicting neprilysin expression in the 4 breast cancer cell lines that I studied compared to normal mammary epithelial cell expression of neprilysin. The 4 breast cancer cell lines showed much lower levels of neprilysin expression than the normal control cell lines, and this was expected based on our hypothesis. P10 qPCR in figure 1B did not reveal the results we were looking for. It only showed upregulation of 2 of the 4 cell lines, and that upregulation was not significant. Ideally, we would like to see upregulation of all 4 cell lines being tested. Using a statistical analysis software, I was able to generate a correlation graph between neprilysin and PTEN mRNA expression levels. In figure 2, it includes all 4 cell lines that were tested, but the correlation coefficient and p-value indicate that there is very little chance that neprilysin is regulating PTEN levels. But an interesting finding that was made is that MDA MB231 cells, which are one of the cell lines that were used in this experiment, do not have any mutations in the PI3 kinase or PTEN, and all the other cell lines that were used do. So when we look in figure 3 at the correlation between neprilysin and PTEN, only observing cell lines that have mutations in PI3K or PTEN, we see that there is a much stronger evidence to suggest that neprilysin is regulating PTEN, and that these results are more likely to be significant, even though they're not significant yet. This insignificance could be due to the low number of cell lines that were used, or the fact that only one biological replicate was used, which would also explain why there are no error bars present in these figures. So based on these results, I was able to conclude that neprilysin was decreased in all cancer cell lines, and these results are consistent with the results obtained by past researchers in this lab, and suggest that low neprilysin expression could be contributing to breast cancer progression. I also found that when looking at specific cancer cell lines that had PI3K or PTEN mutations, there was a negative correlation between neprilysin and PTEN levels. In the near future, I'd like to repeat this experiment using additional breast cancer cell lines that express PI3K or PTEN mutations and see if I was observed the same negative correlation between neprilysin levels and PTEN levels in those cell lines. I would also like to isolate protein and compare levels of neprilysin and PTEN using Western blotting in these same breast cancer cell lines. Long term, our lab would like to further investigate if PTEN and neprilysin correlation is specific to certain breast cancer subtypes, or if this correlation is found in all types of breast cancer. And we would also like to investigate if altering the expression of neprilysin would affect the sensitivity of breast cancer cells to PI3K inhibitors. I'd like to thank the Lipscomb Biology Department for funding my project, and Dr. Beth Conway for her training and allowing me to work in her lab.